So thank you to the organizers for allowing me to speak. And it's always a pleasure to follow up from Dr. Butler. Not only do you get a double dose of Canadian technology, his talk really framed what I'm going to talk about today. As I may have some forward-looking statements, I am required to show this slide. So I lifted this slide from the AACR report in 2015 just to underline the idea that now immunotherapy has been recognized as a potential new pillar for cancer treatment, along with our three standard historical treatments for cancer, as well as precision therapy. I guess I don't need this slide, but really focusing the idea that although we've been enjoying recent clinical success with harnessing the immune response in cancer, it's not a new phenomenon. It can date all the way back to Coley's experiments, and then really culminated with the clinical breakthroughs around checkpoint inhibitors. It's taken a little bit of time to really see the clinical impact of immunotherapies. And really part of that reason is that the role of the immune system in cancer has significantly evolved in the last couple of decades. If you just look at Hanrahan and Weinstein's hallmarks of cancer and how that evolved over a period of a decade to really include the impacts of the immune system in a tumor microenvironment, it really underlines how much we've learned about the role of the immune system. And really the central, and I may be speaking a little bit centric to myself, the role of the T cell in fostering immunotherapy. When you look at key attributes of anti-tumor immunity, really the core of that is generating anti-tumor responses. And although we've had for a long time this appreciation of the central role of the T cell, translating that to the clinic has been a bit of a challenge. We've had significant breakthroughs, which Dr. Butler went over, in modulating some of the effector functions of T cells, but really facilitating getting T cells, generating them to sufficient numbers, and getting them into the tumor has lagged. The classical methods, which have classically been denoted as cancer vaccines, and I'm one of these people who really don't like that term. The term cancer vaccine is a very large bucket, and I don't think it's very descriptive of what we're actually trying to accomplish. I think what we've learned from other therapies, such as adoptive T cell therapy and CARs, is that if you can get enough T cells in the circulation, induced to an appropriate target, we can actually generate clinical responses. But if you look back historically around vaccine technologies, there's been a lot of promise preclinically, but they have lagged in demonstrating clinical responses. So where do we go from here? I think part of what's been established is that the clinical paradigm for immunotherapy historically has followed a very oncology-focused drug development. And there's been a lot of work here, and I'm not going to take any credit for this because it was done well before my time, but what you saw was this really great working group established from the SITC that looked at establishing some new criteria when you're looking at antigen-specific T cell responses. And how do you look at that clinically? When you're looking at inducing antigen-specific T cell responses, there's little need for establishing MTD. It doesn't have that sort of classical dose-dependent toxicity. Dosing and schedule, although very important, isn't determined through toxicity. It really is determined by the generation and the sustaining of immune responses. It's not classically metabolized, so demonstrating classical PK is also a challenge. And there's been a lot of challenges around using RECIST criteria to measure clinical responses, and this has partially been addressed through the iRECIST, but it still has been a real challenge, especially when phase 2 clinical trials are trying to line up those responses to historical data. It has been a very challenging comparison. So really what the focus has been is using proof of principle studies to generate endpoints that both reflect clinical realities as well as biological activity. And really the focus really should be on the mechanism of action of what you're putting into the clinic. So IMV, we've been around for a little while. We really focused our clinical program on demonstrating efficacy that comes around the mechanism of action. So what makes our technology different? I did put in the title fairly provocatively that we have a oncology-driven platform. We've really focused from the beginning of the company, how do we generate T-cell responses that we believe will be sufficient to have clinical impacts in oncology? There's a lot of talk about how antigen is released at the site of injection, and we believe that our technology is the first where it's not released at all. We have a very simple platform. It is oil-based, but it does not include an emulsion. So all the components of the treatment are held within the oil. And so when it's held within the oil, it's actually not released at the site of injection, and it really forces the immune system to take it up actively. So what we see are antigen-presenting cells coming in, taking up the components. This results in a very different kinetics of an immune response, and it also induces very strong immune response, which I'll show you from some of our data. The antigen is then contained at the site of injection for a very long period of time. So this actually changes the kinetics of those immune responses. There's also been a lot of data generated in the last couple of years, mostly around emulsion vaccines, that have called it a T-cell trap or a T-cell graveyard. When you hold the components of the vaccine at the site of injection, they're not available to induce inflammation, and they're also not available to attract T-cells back to the site of injection. So our technology is known as DPX. We like to say we're actually programming T-cell responses in vivo, so to differentiate us from the T-cell technologies that are doing it in vitro. We can do this with both T cells and B cells. I'm just going to talk about our oncology platform today, but we have been also able to leverage this in more classical vaccine approaches to induce B cell responses. It's because the platform itself is fully synthetic, so you can swap out different adjuvants, different components, and really craft it around the immune response that you're trying to generate. And there's a lot of manufacturing advantages to this. It's an off-the-shelf product. It has multi-year stability. It is a very different technology. So our first clinical asset is known as DPX-Survivac. And we're targeting the tumor-associated antigen survivin. Why survivin? A couple of reasons. It is pretty ubiquitously expressed across cancers. It's overexpressed. The antigen is presented by T cells within the tumor. These are just the reported levels of overexpression in some indications. Our lead indication is ovarian, where it's been demonstrated to be overexpressed in over 90% of those tumors. It's an inhibitor of apoptosis protein. It does pretty much what it says it does. It helps cancer cells survive. It is associated with chemoresistance and progression. So as tumors become more chemoresistant, as they progress, they upregulate survivin. And so we, with Survivac, want to leverage the mechanism of action inducing strong T-cell responses and targeting suppression. The actual components are 5 HLA-restricted peptides. So these are minimal peptides. They were originally developed by Merck KGaA as Survivac, delivered in a phase 1 clinical trial, an emulsion vaccine, and they didn't believe that they generated sufficient immune responses to warrant further clinical study. So we brought them in-house and reformulated them in DPX. So our initial preclinical results were that if we reformulate, and this is all in mice, looking at either interferon gamma ELISpot from immune organs or an in vivo CTL assay, when we reformulated these antigens in DPX, we could enhance the development of CD8 positive T-cell responses. And so that really led us into our first clinical indication, which was ovarian cancer. Ovarian cancer is an area that's really ripe for immunotherapy. When you have intratumoral T-cell infiltration in ovarian cancer, it is a positive prognostic factor in that tumor. But really, we believe that inducing a successful immunotherapy would require inducing more CD8 positive T-cell responses. A lot of the currently studied immuno-oncology agents, like checkpoint inhibitors, have not yet demonstrated great clinical responses in ovarian cancer as a monotherapy. So really there's a gap there, and we believe that that gap is inducing and driving T cells into the tumor. And so we have a pretty unique clinical focus. Our focus is we want to show, first off, that our treatment can induce T cells in the majority of patients. We believe that unless you induce an immune response in 85, 90% of your patients, that you don't have a hope in having clinical responses. It's a numbers game. You really need to induce the immune response in everybody, and then there will be a bunch of tumor-specific barriers to getting those T cells out of the blood and into the tumor. So we believe that the baseline has to be that you induce strong, robust T cell responses in nearly everyone. Then you have to demonstrate that those T cells can get out of the blood and into the tumor. And then you'll begin to see your tumor regression. You'll see your objective response rates. And we believe until you show all those, the chances that you're going to have an impact on PFS and OS are going to be minimal. So the one strength that we do have internally is that we like to preclinically model everything we do before we move it into the clinic. So we have some well-established mouse models with what I would call surrogate targets, mostly because our HLA-restricted peptides and survivin are human-specific, making them difficult to study in mouse models. So the first thing we wanted to model is there a way to increase the immune response to DPX-Survivac? And around the time, so this was way back in the early 2010s area, we were like, what can we put with our treatment to really boost that immune response? And around that same time, there had been papers coming out about using very low dose of oral cyclophosphamide. So this is 50 milligrams twice a day, 1 week on, 1 week off. So we call it metronomic intermittent cyclophosphamide. And in those papers, it had demonstrated an impact on Tregs. And so we thought, well, if we can impact the Treg population in the tumor, that would drive the immune response to DPX-Survivac. And so in our preclinical models, we were able to demonstrate that combining the metronomic cyclophosphamide in the drinking water of the animals, and then combining it with DPX, had a significant increase in the efficacy. And then we spent a lot of time picking apart why the metronomic cyclophosphamide had this impact. And what we could show is that it increased the antigen-specific T cells that were available to go into the tumor. So we translated that into a phase 1 clinical trial in advanced ovarian cancer. And I'm going to say, first off, this is in the maintenance setting. So these are in patients who have undergone treatment, who do not have active tumors. We really wanted to look at the immunogenicity of DPX-Survivac in this population with or without metronomic cyclophosphamide. The other thing we really focused on was very intense immune monitoring. We wanted to look at ELISpot responses, tetramer responses, the ability of these T cells to make multiple cytokines or their polyfunctionality, as well as some other markers. So this data was published a couple of years ago. I will first say, because people often ask me this, these are ex vivo ELISpot responses. These cells have not been stimulated, only overnight. So these are pretty strong antigen-specific ELISpot responses in general. What we were really looking at was there an impact of the cyclophosphamide in the advanced ovarian cancer patients? So if you compare cohort A to cohort C, there really was a significant impact of adding the cyclophosphamide on the number of antigen-specific T cells. And we could also demonstrate, if you compare cohort B and cohort C, a dose response. So we dose by volume because it's an oil diluent, that you could see an increase in immune response modulated by dose. We also looked very deeply at the different subtypes of T cells that were induced by the treatment. On the right, you see the tetramers, either ex vivo or after in vitro stimulation. And then on the left, you see some of the different T cell phenotypes that were induced by treatment. And this actually demonstrates it a little bit better. If you compare cohorts B and C, you can see that over time, the development of T cells that are antigen-specific, that are producing multiple cytokines, increases in multiple different T cell types. The one thing we did find is that we sort of used the classical quote-unquote vaccination strategy. We gave 3 doses 3 weeks apart. And one of the things we noticed is that we were treating in the presence of a fairly high immune response. And what that resulted in are site reactions. We started to see reactions at the site of injection. So we followed this up with a phase 1b study to really optimize the dose, because we realized that going forward we were probably going to combine this treatment with another immuno-oncology agent. So in the phase 1b, we tested several different types of cohorts, mostly where we were giving 2 doses to induce an immune response, and then lower doses stretched out over longer periods of time to maintain that immune response. So what we've been able to show is that we could really induce that response in most patients with 2 doses. And then by stretching out lower maintenance doses 6-8 weeks later, we were able to maintain those immune responses for over a year. So really, we're inducing these very strong immune responses that we could maintain for at least a year in these trials. We did have a single case study in this trial of a patient who came in with active tumor. So we were able to follow the impact of the immune response on the tumor in that patient. The study was not powered to look at objective clinical responses, but we did document that in this patient, we saw a shrinking of the tumor by 41% that was maintained for 8 months, correlated with drops in CA-125 and an increased immune response. So it was very exciting for us that we could correlate not only the immune response, but then with an objective clinical response. So hopefully I've convinced you that we believe that DPX-Survivac can induce these strong systemic T-cell responses, and we've come up with an optimized dosing schedule that then could be combined with other immuno-oncology treatments. I don't think I have to convince this audience that really, for immunotherapy, combination is really what's going to have the biggest clinical impact. And ideally, you want these combinations to combine things that induce T-cell activation, that remove inhibitory signals, that supply co-stimulatory signals, and then improve the tumor microenvironment for the immune. So there has been a lot of failures in immuno-oncology, especially around combinations, and that's been particularly evident at this year's ASCO. From the outside, sometimes some of those combinations appeared to be empirically designed. There wasn't a strong preclinical mechanism of action for combining agent A with agent B. Doesn't mean that it doesn't exist, it just wasn't in the literature. So I think we're still in an area where we're going to see more of these failures. And I think once we focus on rational designed combination immuno-oncology that will work from different buckets, then we're actually going to start to see some of the clinical impacts. Our pipeline is pretty clear. This is DPX-Survivac. We have an ongoing trial, which I'll show some of the clinical data from, combining our treatment with epacadostat, which is an IDO1 inhibitor. We have 2 ongoing investigator-initiated trials combining our treatment with pembrolizumab. And we will imminently be starting a basket trial where we'll be combining our treatment with a checkpoint inhibitor. So why epacadostat? Why would we combine our treatment with an IDO1 inhibitor? We have demonstrated that our treatment drives strong T-cell responses. We wanted to focus on something that would function within the tumor microenvironment, and for us that made a rational combination. And when we also had preclinical data, and these are 2 different animal models where we combine DPX, our DPX-Survivac, our DPX, cyclophosphamide, and epacadostat. And so these are just 2 different models where combining the treatments had better efficacy. This was data we presented this year in AACR. And without going into a lot of preclinical data, we drilled down into the tumor microenvironment and really wanted to understand why this treatment was having an impact. And some of the things that we found was that the IDO1 expression on the APCs were increased by our standard treatment, but then were decreased by the addition of the IDO1 inhibitor. The Tregs were actually increased in the spleen, but decreased within the tumors. So we actually see a selective decrease in Tregs. And it increased T-cell infiltration and T-cell proliferation within the tumor. So there's a strong rationale preclinically to why we would want to combine these. And so around the same time that everybody was talking about the the trial with pembrolizumab and epacadostat, we presented at ASCO our preliminary results combining DPX-Survivac, metronomic cyclophosphamide, and epacadostat. So this is preliminary data. It was presented by Dr. Dorigo at ASCO this year. And this trial is almost complete, but these are the preliminary data we presented there. We used two different amounts of epacadostat. The first group is at 100 mg, and the second group is at 300 mg. All of group 1 have been recruited, and this is the preliminary enrollment for group 2. So this is the waterfall plot. The one difference I will say in this trial is that instead of being in a maintenance setting, all the ovarian cancer patients who were enrolled in this trial had to have advancing tumors, either radiologically, which was most of the subjects that enrolled, or also by CA-125. So they all had advancing tumors when they enrolled on this trial. So, so far we have had 4 partial responses, 8 stable disease. So if you think of this, their tumor was advancing and now it has stabilized, and 4 that have had progressive disease. You will see that from the preliminary data, several of the patients that had responses had very durable responses and extended out past the end of the trial. So we are seeing a significant durability. And just to put this in context with other types of immuno-oncology agents in ovarian cancer, you'll see that it is very rare to get objective responses, good objective responses in this patient population. So we're actually very cautiously optimistic about the clinical responses we've seen to date. And we announced after this, at this time, that we are actually extending this trial into a phase 2 where we're actually going to explore the relative benefit of epacadostat by having one cohort with and one cohort without. We also did extensive immune analysis in this patient population. We looked at the immune response in the blood, and all patients that were evaluable had survivin-specific T cells in their blood. We also had a mandatory biopsy. So we had a biopsy before treatment and at day 56, and that was critical data for us around our mechanism of action. We did RNA sequencing, multiplex immunohistochemistry, and multiple measures to really demonstrate that there's something different happening in the tumor after treatment. So I'm going to just show one slide of this data. This is the RNA sequencing data, and what I'm showing you is a T cell pathway analysis. Rather than look at specific markers for T cells, we did complete pathways, and what you're seeing is increases in tumors post-treatment by RNA. So this was confirmed also by multiplex immunohistochemistry. The interesting thing is that both patients in the initial cohort who had a partial response demonstrated increases in T cells in their tumor after treatment. We had one additional subject down here in the corner who also had this evidence of increased T cell infiltration, but progressed through treatment. So we're really, really interested in what was going on with this subject in particular. When we drilled down into her tumor, we found that the patient had evidence of MHC class 1 dysregulation prior to treatment. So this was a patient who was basically exhibited a primary resistance. She could not respond to the peptides in the class 1 restricted peptides in our treatment, which again argued for our mechanism of action, frankly, that we're actually seeing T cells, class 1 restricted, driven into the tumor, and that correlated with responses. So in summary, our phase 1b has evidence of clinical responses in these advanced cancer patients. We had immune analysis showing antigen-specific immune responses. The biopsy confirmed a correlation between induction of T cell responses and our clinical responses, and then a randomized phase 2 is underway. And of course, I did mention that we also have combination trials with pembrolizumab. So what is the support for that? In our original studies, when we looked at the tumors after our treatment, we did see increases in both PD-1 and PD-L1. And so we wanted to understand if adding a checkpoint to our treatment would have increased efficacy. Indeed, it does. These are the survival curves. But what was interesting is that when we looked in the tumors, we actually didn't see any increases in the immunoinfiltration when we use checkpoint inhibitors. So that sort of made us scratch our heads. We assumed that we were going to drive more T cells. There was no more T cells. There were no more antigen-specific T cells in the tumor. So what was the checkpoint inhibitor doing? What it was doing was increasing the cytotoxic gene signature within the tumor. So we're seeing more Tbet, more granzyme, more interferon gamma, but still we weren't really that impressed by what we were seeing in the tumor. So we really wanted to drill down one layer further and said, well, what's happening to the clonality of the T cell response in the tumor when we added a checkpoint? So we used TCR beta sequencing. And what basically we found is that the clonality of the T cell response when we added pembrolizumab was lower. So we were increasing the clonality. We were focusing the immune response to an antigen-specific immune response, which is sort of counterintuitive because there have been reports that adding pembrolizumab would widen, you'd see more epitope spreading. But actually, in the presence of an antigen-specific T cell response, we're actually increasing the clonality of the response, which, if you believe that it's going to be effective, which we saw in these models, it would be. So, and then just as we were presenting our data, there was some data on the effectiveness of pembrolizumab as a monotherapy in a similar clinical population. And as you'll see, it is actually less than 10%. So there is certainly room for adding therapies in this particular indication to hopefully increase the clinical response. So as I mentioned, we have 2 ongoing clinical trials with Merck. They're both investigator-initiated. One is in advanced recurrent ovarian cancer. The other one is in diffuse large B-cell lymphoma. So those are both being conducted independently of the company. And we will follow this up with a trial in 5 additional indications using a checkpoint inhibitor in combination. So what I hope I've convinced you is that DPX-Survivac is a novel T-cell activating treatment, immunotherapy, that can induce robust, de novo, so induce it within the body T-cell responses, that can be sustained at least in ovarian cancer patients. We're inducing T-cell infiltration, and that correlates with the clinical responses we've seen to date. And we really believe that our clinical path is around rationalized combinations based on both clinical and preclinical data. And we believe that we have a platform that can induce a sufficiently robust T-cell response to have an impact on tumors. And I'm happy to take any questions.
Great, thank you very much, Marianne. So I'm going to kick off. So the 5 peptides in the vaccine, the formulation, I should say. Right, the 5 peptides. So what do you think? Obviously, that was in-licensed, as you mentioned, from Merck in the first place. But the original design of those 5 peptides is obviously fairly key to the whole success. What's your thoughts on a widening, a broader set of peptides? Obviously, 5 peptides with different HLA restrictions is still relatively limited. So do you think that there would be improvements if you widened the selection of these peptides?
Certainly. So there's a couple of comments there. One, the 5 HLA restrictions that are included, A1, A2, A3, A24, B7, cover greater than 85% of most populations. So we are getting fairly good coverage, and in our ovarian cancer trial, we don't actually screen for those HLAs, and we have seen off-target immune responses. So we have seen people who weren't classically matched for one of those peptides generate an immune response. So I think as we sort of go along, we're trying to understand some of the additional HLAs we're probably touching. These were all developed by academic groups. 2 of the peptides have been modified to increase their binding to HLA. So, and we always test in our studies the binding of our T cells to both the immunizing peptide as well as the wild type peptide, and we've never seen a difference, something that we've always done. So yeah, I mean, also at AACR, we published that our platform could hold up to 25 different peptides and generate immune responses, more in response to the need of certain other fields that think they need to deliver many, many, many peptides.
It's good. You're thinking that even if you have one peptide for each HLA restriction, then you're also making an assumption that that would be the immunodominant response to that particular peptide.
Yeah, and so we're spending a lot of time understanding the immune responses to different peptides. We're sensitive to the fact that, at any one time we're treating subjects who will not respond to every peptide. So we've had patients matched to 3, but never anybody obviously is going to be matched to 5. So we have to understand the relative contribution of each peptide and the validity of keeping it in the formulation, but it is an active area in which we're looking.
So this is in a lipid nanoparticle, yeah?
Yeah.
And unless I misheard, you said that they were preferentially taken up by the T's and B's?
They're preferentially taken up at the site of injection by antigen-presenting cells.
Okay.
So we believe it's an active process. This is based on data. So what we've done preclinically is we've used MRI technology, supraparamagnetic iron oxide, to tag the antigen, and then look at the kinetics in which it leaves the site of injection and goes to the regional lymph node. It's very different, obviously, from any other formulation we've tested.
Okay, so it's something that is intrinsic to your
Yes.
LNP formulation that they're getting taken up by
As far as we know, we're
specifically?
the only all-oil formulation used to
Got it.
induce immune responses.
Okay.
So you had a lot of preclinical data that metronomic cyclophosphamide effectively reduces Tregs. How about, since you have biopsies in these patients, was that confirmed in the human?
So it was funny. So we did the preclinical studies. We couldn't actually demonstrate. So it had been described in other people that the impact was on Tregs. What we saw when we did it in preclinical studies was that it increased antigen-specific T cells, but we couldn't demonstrate in the tumors that it was specifically Tregs. And then we now have our biopsy tissue, and we haven't seen a lot of Treg depletion. So the problem with cyclophosphamide is it has been described to have pleiotropic effects. What we've been able to demonstrate is that it increases antigen-specific T cells, likely by dampening some suppressive mechanism, but we've never caught it in any of our sampling to say, okay, it's definitely that cell.
Hey, I got two quick questions.
Yeah.
The first one is, does the site of injection matter, like where you injected the therapeutic?
So we don't necessarily think so. So one of the things I didn't talk about in our phase 1b is that mostly for aesthetic reasons, in ovarian cancer, we inject in the legs. We did test injecting all the ones in a single site versus going to the other side, and that didn't seem to have an impact. We have tossed around the idea, do we want to generate T cells in the local regional area? And I think that's something we'll explore when we go into different indications. Right now, because most of our data is in a single indication, it's hard to say, but it's something that we continually look at. In our models, mice lie. So it's not something we've ever seen a difference in mice, but as we go into additional indications, it's something we'll look at.
And one more thing. I was wondering if I know you showed that there was different populations of T cells that were, you could see,
Yeah.
but in long-term injections and constantly experiencing the antigen, did you see an increase in the effector memory cell populations?
So there's something that
long
now
term?
that we have much later. So in our current trial, we have patients who are having clinical responses out to a year and plus, and so we're spending a lot of time really characterizing those T cells, and it's not data we've talked about yet, but we're looking at things like, are they still effector memory? Are we seeing any indication of exhaustion, which we haven't yet, and it may be just how the antigen is experienced that it's such a low dose over a period of time that we're not seeing it.
So long-term exposure to antigens, like a late antigen, I guess you could say,
Yeah.
does increase the presence of these CD4 memory populations. They tend to require that phenotype.
Yeah.
So I'm just curious to see what happens to your program.
So luckily, we take 11 samples from each of our subjects in our current trials. So we take it over time, and then if they extend on into treatment, we'll take additional samples. So we're now just getting the samples at sufficient time points to start to answer those questions.
Hi. In terms of immune monitoring, you mentioned that all the patients developed T cell, antigen-specific T cells, but clinically you see a range of responses. So is there a correlation between the amount of T cell response or the breadth of T cell response and the clinical response?
So currently, what we're seeing is that the barrier, if you want to give it, is if the T cells leave the blood and go into the tumor. So all the partial responses we've had, we've demonstrated that the T cells can leave the blood and go into the tumor. It becomes very difficult then to correlate it to the systemic immune response. Indeed, anecdotally, we see the patients who have the best response, their blood response actually goes down, which you think if they're leaving the blood and going into the tumor, what's circulating or available to circulate will be less. So I think that's why I said I think you have to induce an immune response in almost everybody, because if you don't, you're playing the numbers game. Right now, not everybody, all those T cells are going to have other barriers. So unless you induce an immune response in everyone, it's going to be very difficult to see those clinical responses because of the other things. And I think as we get more data and we understand more, we may be able to manipulate. We spend a lot of time picking apart those biopsies and what's the barrier. We don't have an answer yet.
So the trafficking of the T cells to the tumor is the key.
Is the key, yeah.
Well, thank you very much indeed, Marianne.
Thank you.